This video covers false prediction number 10, lineage-specific biology. So look at this lineage in blue. It has 5 species in existence today. Now, according to evolutionary theory, these 5 species share a long evolutionary history. They have only been evolving independently for a relatively short amount of time. Zooming in on these 5 species, so there's going to be differences between these species. If there weren't differences, they wouldn't be different species. But the differences are limited. So why is that? Well, there's a limited time since that common ancestor. There's only so much time for differences to evolve. They can't be very different. Any evolution that happens that has a deleterious mutation isn't going to work very well. So there's constraints on what sort of evolutionary changes can happen. There's a ratchet effect. So all that history of common evolutionary history, that there's a direction to evolution. It's built up the genome, the organs, all of the different aspects of the body has been built up, and then you've only had a little bit of time of independent evolution. Now you can't just go back and say, oh, I'm going to switch out the genome. I'm going to put in a whole new genome. You can't just go and put in a whole new brain. You can't switch out the heart. So the evolutionists talk about this ratchet effect, that there's limitations on what evolution can do once things get burned in, so to speak. And then, of course, jumps are not allowed in evolutionary theory. Things have to be gradual. So there's a lot of limitations on what can happen. So what about a lineage-specific design? What if there is some design that's lineage-specific, like in this turquoise lineage, it has something that the others don't have? Well, it must be limited. That is the prediction. Biology is not significantly lineage specific. Yes, there are differences between lineages, but it can't just have significant radical lineage specific designs. That's the prediction that evolution makes. It's very natural. This comes right out of evolutionary theory. It is false. It's contradicted by a wealth of scientific evidence. If you talked about lineage-specific designs, say, 50 years ago, 100 years ago, back in the mid-20th century, people would look at you funny, like, what are you talking about? It was not a term that had any currency. But about 20 years ago or so, that term finally broke through. The evidence was just too strong, and so it's now become a key word, not because of evolution, but in spite of evolution. It runs throughout biology, as we're going to see in this false prediction number 10. So you have papers out that talk about lineage-specific this, species-specific that, lineage-specific, and so forth. It's easy to find this now in the literature. What I want to do here is just show you 3 examples to give you an idea of what kinds of lineage-specific designs biology has. I began with a lot more examples. I had a dozen examples that I was looking at. I was going to base this video on, but it would have been too long of a video. So my point is, it's not difficult to find these examples. It's not a stretch. I'm not digging around to find examples of lineage-specific designs. Let's look at 3 examples: germlines, dinoflagellates, and the Kinetoplastid. So germlines, what's that? So when you have a sperm and you have an egg and they get together, that creates the zygote. That's the single cell that starts a new individual. The zygote divides into daughter cells, and those daughter cells divide and so forth in the embryonic development pathway, where you're growing the individual, right? Well, there's something else that happens. Not only is this young individual growing and getting bigger, but something else happens. These primordial germ cells, or PGCs, are created, and they give rise to a germline. The germline ends up producing, well, if it's a male, the germline ends up producing the sperm. If it's a female, the germline ends up producing the egg. The germlines first emerge in early embryogenesis with the formation of PGCs. So the germline is fundamental. It's core to the cycle of life. It completes this cycle. Germ cells have been considered the ultimate stem cell because they are able to give rise to all organismal cell types. Germlines are the cell lineages that give rise to the sperm and eggs in animals. The germlines first arise from primordial germ cells during embryogenesis. Germlines are the specialized cell lineage contained in the gonads of sexually reproducing animals that give rise to the sperm and eggs. The germ cell lineages are separate from the soma and are the only source of heritable genetic variation passed between generations, providing them with a crucial role in reproductive success, fitness, and evolutionary biology. When you have any design that shows up in a large group, well, it's going to be, according to evolutionary theory, in the ancestors as well. So according to evolution, their ancestors shared this design as well. So I draw the yellow circles throughout the tree. This design was present in the previous ancestors, going back to the ancestor of all the sexually reproducing animals, according to evolutionary theory. That's the way it has to work. But the germline designs are lineage specific. Yes, all of these animals have a germline, but it turns out there are different germline designs. For example, PGCs may emerge via two very different modes, induction and inheritance. They are lineage specific. In other words, you see the inheritance and the inductance modes in different species, which they're not clustered, they're just different species have these designs. If you're an evolutionist, you'd like to see them clustered. So here's a hypothetical clustered distribution. And so if you're an evolutionist, you could say, well, yes, you see, originally it was red, maybe that's the induction design, and then there was a point where the inheritance mode evolved, and then all the subsequent animals had inheritance on that branch. But it isn't like that. So often it is the case where they're not clustered. It's like this. So let's look at an example from insects. So this evolutionary tree of insect groups shows that on the right, you have different groups of insects. The red boxes represent the induction mode, but the green boxes represent the inheritance mode. So here you see how they're kind of interspersed. They aren't clustered. So according to evolutionary theory, well, you have it beginning in the lower left, the common ancestor has the induction mode, but then there with the holometabola, the inheritance mode evolves. And then after that, the species, you have the inheritance mode, except, wait a minute, there were three lineages that evolved back to the induction mode. And so it's just a just-so story. You just have these evolutionary events hypothesized in order to describe the distribution that's observed today. So to rationalize this, evolutionists will appeal to some sort of advantage. The inheritance mode evolved because of some sort of fitness advantage. Since production of the germline is a critical event in development and evolution, it is surprising that dramatic changes in how this cell fate is established have occurred several times in insect evolution. So, right, it is surprising according to evolutionary theory. The question of why evolution would switch modes is, quote, difficult to address. Yes, it is a difficult problem. So they're going to appeal to benefits, selection benefits. In other words, since the inheritance mode evolved, it must have some selective benefit. So that explains why it evolved. So ultimately, you get into this sort of circular, just-so story sort of explanation, appealing to selection so that therefore it evolved. So that's the argument, but then, well, if the inheritance mode had those benefits, why then did the induction mode reappear later on in these three groups of insects? Well, in that case, the inheritance mode wasn't so good. So basically, it's special pleading. This is the fallacy of special pleading. You're simply describing whatever you observe according to the theory. And evolution shifts back and forth between these fundamental designs very easily. But it gets worse because it isn't just insects. I've shown you that example. Crickets, mice, and salamanders, for example, have the induction mode. Flies, wasps, nematodes, and frogs have the inheritance mode. So these They're not clustered. They're just kind of sprinkled throughout the evolutionary tree. They are lineage specific. So these are more lineage specific examples of this fundamental germline that just shows up with different groups of species. But again, it gets even worse because even within a mode, the designs are lineage specific. While the inheritance mode is shared between flies and nematodes, the key upstream regulators appear to have arisen independently and in a lineage-restricted manner in these two model organisms. So if this is even more lineage-specific designs. This study suggested those genes were increased targets of short-term selective sweeps. Now, I hope a red flag goes up for you there. So this is teleological language. This study suggested those genes were increased targets of short-term selective sweeps. In other words, evolution has targets, and it performs selective sweeps. This lineage-restricted de novo gene has evolved crucial functions. This is another interesting feature. It's not that new genes have evolved and they just happen to be kind of helping out. I mean, these are performing crucial functions. So that's another problem, this ratchet effect I talked about earlier. It's very difficult for evolution to rationalize. Remarkably, our data here show that these two genes, which are de novo, have arisen independently in the worm and nematode and fly, respectively. We conclude this could be a common property of de novo germplasm genes. In other words, you have these new genes, these gene differences. If evolution is true, they must have arisen rapidly. They diverged and new genes arose rapidly. And so we conclude that this could be a common property. In other words, for some unknown reason, de novo germplasm genes in particular evolve rapidly. But there's no actual scientific evidence for this. This is simply a conclusion to save the theory. If evolution is true, they must have evolved rapidly. So I guess that's just the way these sorts of genes work. So germlines are fundamental in the development of animals. Germlines arise in two different modes, and they are lineage specific. Each mode has lineage specific details as well. That are so within each mode you have more lineage-specific designs, and there's no smoking gun. What do I mean by smoking gun? So this is an important concept. When you have a false prediction, what you want to have is that it reveals something. You learn something about the theory, and it's an aha moment. Oh, gee, we didn't think of this at all, but now that we see this false prediction, we realize if we just tweak the theory, change the theory around, we can not only explain this false prediction, but a whole host of other issues that we had. This would be a great outcome of a false prediction, but that's not the case. What we have here is ad hoc, just so stories explaining the evidence that we have, these germline modes and the lineage-specific designs. Okay, that's germlines. Let's move on to dinoflagellate genome. So let's start off with a press release. Dinoflagellate genome structure is unlike any other known. Here is a couple of images. The dinoflagellates are a single cell eukaryote. Some of them are found in freshwater environments, others in marine environments. Some are parasitical, many are symbiotic. But what's really interesting is you go into the nucleus and look at their genome, and you see something very different from what is typical of eukaryotes. Rather than the flexible X-shaped chromosomes familiar to humans, dinoflagellates organize their genetic material in orderly blocks along rigid rod-shaped chromosomes. Dinoflagellates are eukaryotes, but unlike the chromosomes found in humans, which form an X shape, dinoflagellate chromosomes assemble in a straight rod-shaped structures. Genes line up in blocks along these rods, with each block oriented in the opposite direction of its neighbors. A block's orientation dictates which direction the cell can read the genetic instructions. This unusual alternating structure both influences the overall shape of the chromosome and likely regulates how and when specific genes can be accessed. Dinoflagellates quote, don't fit with everything else we know about eukaryotes. How they structure their chromosomes, how they structure their genomes, how they regulate transcription. Indeed. Furthermore, the team found that pairs of neighboring blocks tend to interact with each other, while faraway blocks rarely do so. While both neighboring blocks untwist during transcription, granting access to their genetic material, blocks outside of that pair remain rigid and unchanged. This finding hints that some kind of barrier exists between the different block pairs, and that the barrier must be something really important in organizing the chromosome. Okay, so here are some journal papers. This paper looked at 15 different Dinoflagellate species and their genomes, and they inferred these protein-coding genes and compared them across these species, and here's what they found. Remarkably, predicted proteins coded by the vast majority of these conserved lineage-specific genes do not share significant sequence similarity to any protein sequences in the UniProt database. Yes, this is remarkable. In other words, you've got these 15 dinoflagellates. They collect all of the likely protein coding sequences from them. They do a all-against-all search to find similar homologs. What other species, what other genomes in nature have protein coding genes that are similar, and they don't find hardly any. That's the vast majority of these protein coding genes in these 15 dinoflagellates are not found anywhere else. This is tens of thousands of genes without homologs outside of the dinoflagellates. This is a remarkable finding. This is lineage-specific design on steroids. Some families of dark genes appear to be specific to some isolates, so unique to this particular species. And this chart, I won't go into the details here, but this chart illustrates some of these findings. You can see along the bottom there that you're looking at tens of thousands of lineage-specific gene families. This is radically lineage-specific. Both the genome structure and the information, the content, the genetic content of the genome is radically lineage-specific. And that's what this paper concludes, paraphrasing, our results reveal lineage-specific, quote, sequence and structure. Dinoflagellates seem to defy many of the cellular features found in other eukaryotes. Most interestingly, dinoflagellates fold their chromosomes in a way that is distinct from other eukaryotes, and that is also distinct from prokaryotes. Yes, so this is lineage specific, but it's also a case where you miss a opportunity for a smoking gun. What if the dinoflagellates kind of looked like the prokaryotes? That would have been a smoking gun. That would have been a very interesting piece of evidence. It would have suggested that this dinoflagellate design was kind of the transition between prokaryotes and eukaryotes. That would have been a very interesting finding. It isn't. This dinoflagellate genome is nothing like the prokaryotes. One principal hallmark of dinoflagellate chromatin is the observation that most of their DNA is not wrapped around nucleosomes. These chromosomes appear as permanently condensed rods. It's very different, very lineage specific. Dinoflagellate chromosomes represent a unique evolutionary experiment. Okay, that's another red flag for you. Evolution conducts experiments, according to this paper. Dinoflagellates possess numerous highly divergent molecular features, including uniquely among eukaryotes, the loss of nucleosomal packaging of chromatin. Now, it says here the loss of nucleosomal packaging. There's no evidence, no scientific evidence for any loss. They don't have nucleosomes. That's what the finding is. That's an evolutionary gloss, that it was lost. They pose intriguing questions regarding the adaptation of transcriptional and regulatory mechanisms. In other words, how did they evolve? Yes, that is an intriguing question. Okay, so let's summarize the dinoflagellate genome. The eukaryote genome is in the cell nucleus. There are several hallmarks to the design that are consistently found, but dinoflagellates are a stark exception. Unlike even its close neighbors, it is uniquely different. It is lineage specific. The genome structure, shape, packing, sequence, and regulation are all strikingly different. It is a radically lineage specific design or set of designs. And it's not like the prokaryotes, so we don't see a smoking gun there either. Okay, so that's the dinoflagellate genome. Let's move on to the Kinetoplastia. This is a phylogeny of the single-cell eukaryotes. We just looked at the dinoflagellates, which are over there in the alveolates. Now let's look at the Kinetoplastids, which are over in the excavates. I'm showing an image there of the Trypanosoma brucei, which is a dangerous parasite that has been heavily studied. We'll look more at that. So first, let's look at a textbook eukaryote cell. This is a plant cell. It has a nucleus. The nucleus is rather large. It also has a number of mitochondria, which are smaller, and has a simple, small genome compared to the more complex genome in the nucleus. All of these rules are broken in the Kinetoplastia and broken in very significant ways. So here's a rendering of that Trypanosoma brucei, and you can see the nucleus there. You also can see the mitochondrion, but it's not mitochondria in this case. You'll notice that it's mitochondrion. Why is that? Because there's only one. The Kinetoplastia have only one mitochondrion, and it's rather large also. And it has a very interesting genome. It's called the Kinetoplast DNA, or kDNA. So you can see kDNA there. And it's attached to the flagella. And you can see the flagella, that gray line, a wiggly line there. So that's the flagella outside of the body of the Trypanosoma brucei. So there's one mitochondria, not many. It's big, not small, and it has a large complex genome. It's called the Kinetoplast DNA, or kDNA. It's often shaped like a disc, and it is large, and it is very complicated. Here's an image of the kDNA. This is from Crithidia fasciculata. Okay, so here's this figure expanded here showing the rendering of this disc. There's this kDNA disc. So in other words, the genome is shaped like a hockey puck, a disc. This disc is made of thousands of small DNA circles called mini-circles and dozens of larger DNA circles called maxi-circles. The mini and maxi-circles are packed into the disc. They unfold when removed from the cell. So when you remove them from the cell, they kind of unfold and flop open into a sheet, a two-dimensional sheet that's like chainmail. These circles, these mini circles, are catenated. They're networked. They're interlocked. They're chained to each other. Somehow these thousands and thousands of these mini circles have been woven through each other. So you have this chainmail sheet that's all been packed up into this disc. It's a phenomenal design. And so here's this shown in this rendering. You can see these mini circles on the order of 5,000 mini circles in the lower right there, intercatenated network, and then you've got some maxi circles as well. Interesting. Kinetoplastids usually have their mitochondrial DNA organized in a unique eponymous structure termed the kinetoplast DNA, which is composed of a densely packed network of thousands of mutually interlocked circular DNA molecules. Interestingly, kinetoplastids have a single large mitochondrion with a disc-shaped structure called kinetoplast. This structure has a complex network of concatenated DNA rings. Kinetoplast DNA is composed of thousands of circular molecules topologically interlocked to form a single network. The most remarkable feature of kDNA is that all of the mini and maxi circles are topologically linked to form a single planar network In situ, the Trypanosoma brucei kDNA network is condensed into a compact disc, approximately 100 nanometers thick and 650 nanometers in diameter, which is anchored to the base of the flagellum. Back to our rendering here. You can see how the kDNA, the hockey puck, the disc, is attached to the flagellum. Unlike any other DNA in nature, kDNA is a network containing thousands of DNA minicircles topologically interlocked, lineage specific, unlike any other DNA in nature, even its neighboring lineages. The isolated networks, which resemble medieval chainmail, are easily viewed by electron microscopy. The highly unusual kDNA comprises a network of two genetic elements, the maxi and mini circles, which are topologically interlocked, forming a disc-like structure. This is radically different from textbook eukaryote cells. But what are the mini and maxi circles used for? Well, so here's a chart of how the maxi circle and mini circles work together. Let me just not go through that in detail, but zoom in on the maxi circle sequences. So the maxi circle in this case is shown to have 20 genes. But wait, of those 20 genes, what is this? 12 are cryptogenes. What's that? Cryptogenes must be unencrypted. They must be edited to unencrypt them. As they are, when they are first transcribed, they're unusable. So that's where the minicircles come in. The minicircles are used in a massive editing process. So here is a model of that editing process. So let's start with a maxi circle, and okay, let's take a gene, and this would be an encrypted gene. So you transcribe it, you have a copy of that gene. It's called mRNA, although it's also called pre-mRNA because, like I said, it's unusable when it's initially transcribed since it's an encrypted gene. And then you have some mini circles, and they have some segments that are going to be transcribed. What do? Well, they're called guide RNAs or gRNAs. It edits that orange part. So I'm drawing that part orange to show that it's been edited. Here is a second gRNA now. Now, where does it go? You would think that, well, it probably goes here, right? No, it does not go here. It goes here. There's an overlap. It overlaps with the first segment that was edited. Why is that? Why does it overlap? Because it binds to the tail end of that previously edited segment. So that overlap region, you have good binding. The overlaps are coordinated. The gRNAs know about each other. The gRNAs are programmed to work together. Each gRNA directs edits that generate the anchor region for the next gRNA. Thus, the RNA editing process is sequentially dependent on correct editing by each gRNA. In massively edited mRNAs, editing events proceed sequentially along the mRNA and require multiple overlapping gRNAs. To produce functional mRNAs, a multitude of nuclear-encoded factors, protein machines or molecular machines, mediate interactions of maxi-circle-encoded pre-mRNAs with a vast repertoire of mini-circle-encoded guide RNAs. Trypanosoma brucei, as other Kinetoplastids, has a complex mechanism of editing mitochondrial mRNAs that requires guide RNAs coded in the DNA minicircles in the kinetoplast. This is radically different, and as I hope you can see, extremely complicated, and it's radically different from textbook eukaryotes. Now, what about replication of all this? Because in cell division, everything needs to be duplicated, and these single-cell eukaryotes are going to have to divide. They're going to have to replicate, including the mitochondria and its genome. That's got to be duplicated. So how are you going to do that? Mini circles decatenate from the network, and replication can take place. So you're going to cleave those, decatenate them, and make a copy. The replicated many circles then reattach. The trypanosoma mitochondrion requires a highly precise genome segregation system. Yes, this is very complicated. In this review, we focus on this system termed the TAC, a fascinating structure exclusively found in Kinetoplastia. Determining how such an intricate network of two intercatenated genetic elements replicates is a formidable problem. Thus far, more than 25 different proteins have been identified that contribute to this process. Currently, more than 30 proteins have been described to be involved in kDNA maintenance, including replication, while far fewer are required for this process in other eukaryotic systems. Very different here. This review compiles 74 processing enzymes, RNA binding proteins, and factors with unknown functionality that nonetheless are associated with RNA processing complexes. There are a lot of molecular machines at work doing all of this job here, and many of them are lineage specific. Another interesting feature of kDNA replication is that the pattern of replicated DNA in the network suggests what? That the kinetoplast disk as a whole undergoes a rotational movement. I would have just said it rotates, but in any case, this is a fascinating finding. Now, this isn't true for all of these species, but it varies at least. The dynamics of the rotation vary, but there's evidence that this kinetoplast disk, this hockey puck, is rotating as the replication process is happening. Fascinating. However, there's no motor no known motor to execute this rotation. Okay, moving on. So back to our drawing here. You can just imagine this sort of a complex process where you're going to not only make a copy of all these things, but then you're going to spin a new one and build up somehow. You know, we talked earlier about how when you unfold this thing, it's this huge chainmail structure, and somehow that's all folded in there. Fascinating. So the summary so far. We looked at the Kinetoplastia lineage and its unusual mitochondrion. There's only one. It's very large, has a very different complex genome structure, the kDNA. It has a unique mRNA editing process. It also has a unique replication process using this TAC system, another lineage-specific structure. And it looks like the genome disk is rotating. So this is radically different from textbook eukaryotes. And it's, I hope you can see, very complicated. The kinetoplastid mitochondrial transcriptome is exceedingly complex and contains RNAs transcribed from two types of mitochondrial circular DNA molecules, maxicircles and minicircles. The mitochondrial DNA of trypanosomatid protozoa consists of a complex, intercatenated network of tens of maxicircles and thousands of minicircles. Kinetoplast DNA is the most structurally complex mitochondrial DNA in nature. And remarkably, some of the protein coding genes are encrypted. The kinetoplast is a specialized region of the mitochondria of trypanosomatids that harbors the most complex and unusual mitochondrial DNA found in nature. Now here's an interesting quote. Life is characterized by its vast diversity. Yet most experimental research in eukaryotic biochemistry and cell biology has focused on a limited number of model systems, such as yeast and mammals. Such a myopic view of eukaryotic diversity hardly scratches the surface of the true breadth of molecular complexity. Of course, all of this, as I have said, is lineage and it's species specific. The diversity of cellular and molecular systems in microbial eukaryotes is simply staggering. Kinetoplastids are notable for doing things differently. A radical and unique type of RNA editing occurs in the mitochondria of the kinetoplastid protozoa. Both euglenozoans and alveolates have a reputation for doing things their own way, which is to say that they have developed seemingly unique ways to develop important cellular structures or carry out molecular tasks critical for their survival. Now, this is not little side tasks. These are crucial tasks and they're doing them their own way, unique ways. So this is lineage specific. Narrow phylogenetic distribution. When you see that phrase, narrow phylogenetic distribution, that is kind of a synonym for lineage specific. It's narrow in the phylogenetic distribution. It occurs only in this one place, this one lineage. It's narrow. So that means it does not come from some common ancestor way back in the evolutionary tree. It must have evolved. They use the word derived. It must have, according to evolutionary theory, it must have evolved within that lineage. This raises intriguing questions of how and why it evolved. Yes, indeed. Again, the narrow phylogenetic distribution, it's lineage specific. The Kinetoplastids are a group of unicellular flagellates of the phylum Euglenozoa that possess many particularities related to their cell biology, biochemistry, and gene expression. So again, particularities, it's lineage specific. But it's more than just lineage specific, because within the Kinetoplastia, there are differences. So it's species specific. Maxicircle and minicircle sizes vary among different Kinetoplastids. The specific mRNAs that undergo editing and the extent of their editing vary among the different Kinetoplastids. Our laboratories and others have found that kDNA actually exists in a wonderful diversity of structures. Okay, so how did these species-specific designs evolve? Why such hotspots for the evolution of novel solutions to problems should exist in the tree of life is not entirely clear. I think that's an understatement. It definitely is not entirely clear. Different evolutionary hypotheses have been proposed to explain the origin and persistence of this expensive editing system, although no consensus has been achieved yet. mRNA editing in Kinetoplastids is an intriguing evolutionary mechanism. Questions about how and why such complex and energy-expensive mechanism has evolved are still controversial. Why did natural selection not erase such complex machinery? The derived nature, again, that word derived, this means it is lineage specific. The derived nature of RNA editing raises intriguing questions about how and why RNA editing systems arise and how they became fixed as additional essential steps in genetic information transfer. At present, we can only speculate about the evolutionary forces driving the Kinetoplastid and diplonemid protists on the path toward giant non-nuclear DNA contents. Non-nuclear meaning they're not in the nucleus, they're in the mitochondria. And it is a huge genome that's packed into that disk, especially given that their sister group, the euglenids, did not follow this path. An accurate perspective of the kDNA expansion and evolution of their unique editing across Kinetoplastids has been difficult to achieve, yes. The origins of the gRNA sequences and their utilization in editing is a fascinating mystery. One hypothesis is that gRNAs evolved from duplicated and repackaged elements of ancient correctly encoded precursors of the current cryptogenes. For various reasons, that hypothesis is questioned. That's kind of the story for all of the different hypotheses. There is a raft of ideas that have been thrown around, and none of them work. So that's why there's so many. There's no one good explanation. There are many hypotheses. The complexity of this editing process, combined with its apparent confinement to one organelle and one group of organisms, has fueled multiple ideas about why it exists in nature. Multiple ideas. Several attempts to answer such questions were made. So a lot of different ideas have been thrown around. Many of them depend on serendipity. It just so happened. An alternative view is that the RNA editing apparatus already existed, potentially serving a different purpose altogether, and was ultimately fixed within the cell through fortuitous events. So now they're really heaping on the serendipity. Yeah, we already had all these systems, these molecular machines doing other things, and now they just fortuitously got together and formed this new incredible capability. That's about the level of these explanations. They are ad hoc, just so stories. It just happened to happen. Another is that this editing could serve as a mechanism of change to more rapidly evolve mitochondrial genome output, leading to greater evolvability for an organism. Versus, hence, the RNA editing process may operate as a proofreading system to weed out mutations by making them lethal. That's reducing the evolvability. As the extensively overlapped genes share most gRNAs, this strategy would ensure that almost all of the genetic material is protected. It's against evolvability. So two conflicting hypotheses here. They can't both be right. Many hypotheses are teleological. I pointed out a few examples along the way, but here's some more. In other words, during their long evolutionary histories, protists have solved general biological problems. I hope you see the teleology there. In evolution, species solve problems. Under the constructive neutral evolution scenario, existing and then increasingly specialized machinery emerged to counter an original mutation, followed by subsequent ones. So whenever you see that infinitive form, that's a tip-off to counter. So in this case, evolution creates machinery to counter mutations. That is teleological. Final causes, Aristotelian teleology. The origins and evolution of editing are entwined with the possibility that organisms are currently exploiting editing as a regulatory mechanism. So in evolution, organisms exploit mRNA editing. This system probably arose in an ancestor of the trypanosomatids as a molecular mechanism designed to avoid mini-circle losses by missegregation. Designed to avoid. There's kind of a double hit there. You've got the infinitive form, to avoid, and then you've got designed. So in evolution, catenation arose as a mechanism designed to avoid losses. Very teleological. RNA editing appears to have arisen in evolution multiple times in different organisms as a way to correct errors and modulate genetic sequences. So in evolution, mRNA editing arose to correct errors. To provide a solution, kDNA formed to provide a solution to segregation. The single nucleoid nature of the kDNA requires a highly precise mitochondrial genome segregation system. This led to the evolution of the TAC. So evolution meets requirements. You give it requirements, it's going to meet those requirements. Teleology. As the extensively overlapped genes share most gRNAs, this strategy would ensure that almost all of the genetic material is protected. So evolution has a strategy. Okay, so to summarize, the Kinoplastia. It has an unusual mitochondria, very different complex genome, the kinetoplast DNA. It has a unique mRNA editing system. The gRNAs are used from the DNA minicircles. It has a unique replication system, the TAC, and the rotating genome disk. It is radically different, unique, and lineage specific. It is highly complicated, and the origin hypotheses get complicated, and they are speculative and ad hoc. Teleology runs through the literature. Okay, so we've seen these contradictions. We've seen some examples here, 3 examples of contradictions of this prediction number 10. So what's the summary if you had to put it on one slide? Biology has significant and radical lineage-specific designs contradicting evolution. So what about the outcome? How do evolutionists respond to this? Well, along the way, I've pointed out quite a few responses and how evolutionists have responded to this failure of this prediction. So I won't go into too much detail here, but just to summarize, this false prediction has not led to a smoking gun or otherwise to scientific breakthroughs. It has led to many evolutionary hypotheses. There's no single compelling hypothesis. Many hypotheses are ad hoc or just so stories. And just as an example here, one that we saw earlier, quote, the RNA editing apparatus already existed and was ultimately fixed within the cell through fortuitous events. So that's just an ad hoc explanation. We're not getting into the details. What mutations happened when? What's the probability? Why should we believe this? None of that. You don't see that. Many of the hypotheses are underwritten by teleology. So that's the summary. So we've seen the prediction, we've seen the falsification, and we've seen the response. What about the failed theory virtues that we've been looking at? Evidential accuracy, causal adequacy, internal consistency, internal coherence, universal coherence, simplicity, durability, and fruitfulness. How, in the light of this false prediction number 10, how would you judge, how would you evaluate evolutionary theory? So on the first one, evidential accuracy, well, the failed prediction is itself a failure of evidential accuracy. For evidential accuracy, you want to have the theory fit the empirical evidence well. Well, it didn't do that. It failed. So you fail on the evidential accuracy. What about causal adequacy? The theory's causal factors plausibly produce the effects in need of explanation. Well, that one has failed too. We looked at the different ideas on, how could evolution create these things? How could these have evolved? And the fact that there's so many explanations, and because there's no one compelling explanation, shows that there's no causal adequacy. And the fact that the explanations themselves are ad hoc at a high level and often are just so stories and are underwritten by teleology. You don't have a detailed accounting of just exactly what does evolution do here. Instead, it's, well, this appeared and that appeared and this would have benefits, so it evolved, and so forth. So you don't have causal adequacy. What about internal consistency? The theory's components are not contradictory. Well, we just saw one example a few slides back on the evolvability. One explanation calls for a reduction in evolvability, another one calls for an increase in evolvability. So right there you have a contradiction. But more fundamentally, a larger problem that resurfaces more or less continually with these false predictions is evolution. The theory of evolution is fundamental to the theory that there's no teleology. The theory is just natural laws acting blindly, and then voila, the entire biological world appeared. It took time and so forth, of course. I don't mean to understate that, but it is. We need to understand you cannot have teleology, you cannot have final causes. There's no goals or objectives. You cannot have that. You cannot induce the right mutations to happen in evolutionary theory. Mutations have to be a biological variation of any sort, but mutations is usually the sort, must be blind to need. Evolutionary change must be just random with respect to need. You cannot have teleology. Things don't happen because of this, because of that, to meet this goal, to meet that goal. That is completely out of bounds. And so we see that over and over in the literature. So there is a massive internal contradiction going on that shows that the theory fails on internal consistency on this false prediction as well as many of the others. What about internal coherence? The theory's components are coordinated into an intuitively plausible whole. The theory lacks ad hoc hypotheses. Well, no, we saw ad hoc hypotheses in response to this false prediction over and over, repeatedly. Ad hoc hypotheses that invoked serendipity and so forth. So no, we don't have that. It fails on internal coherence. What about universal coherence? The theory sits well with other warranted beliefs. Well, what are the other warranted beliefs? Things like physics, statistical mechanics, statistics, molecular biology, and so forth. Those other known, well-established sciences are not consistent with evolution's claims here that these random mutations created such complexity. You just have these massive contradictions between evolutionary theory and the other branches of science. So there is not a universal coherence. There is a conflict. What about simplicity? The theory explains the same facts as rivals, but with less theoretical content. No, that's another aspect of the hypotheses that are set forth to explain these false predictions. The hypotheses are increasingly complicated. You're adding more to the theory. You're patching on. You don't have these smoking gun solutions where, everything falls into place. Now things become simpler. You don't have that. You have increasingly complex explanations. It fails on simplicity. What about durability? T has survived testing by successful predictions. Well, no, we're seeing one false prediction after another. These are fundamental predictions to the theory, and it's failing over and over, so it's not durable. And finally, what about fruitfulness? Is it fruitful? Did this false prediction number 10 lead to a smoking gun or otherwise to theoretical elaboration, non-ad hoc theoretical elaboration, novel predictions, unifications, and so forth? Did it lead to new scientific breakthroughs, new scientific discoveries, new scientific knowledge? Was it fruitful? No, it was not fruitful. You can look at all of the findings in this research, and none of them depend on evolution. The studies are studies of how does this work? How do the Kinetoplastid, the Dinoflagellate, the Germlines, how do they work? How do these different contradictions to this prediction, how does biology work? Evolution was redundant, it was gratuitous, it was not needed to discover any of these findings. Now, sometimes this isn't true. We've seen some false predictions where, the desire to figure out how evolution did this led to some findings. So these false predictions don't always portend failure across the board. Sometimes you see little points of hope that any theory, no matter how false it is, can have virtues now and then. But often case, and I think in this case, evolution fails on all of these theory virtues on this false prediction number 10. So I give it the check on that it fails on all of these. Religion drives science, and it matters.